Hello friends, today we will discuss about lysosomes. See, the lysosomes literally means lyso means breaking and soma means body. So the lysosomes contains various types of hydrolytic enzymes, and that's why they break down each and every macromolecule that is found in our cell. So that's why they are named as a lyso, that means breaking, and soma means body. Now, these lysosomes, they were first discovered by the Christian de Douai, a Belgian cytologist, and for that purpose, he got the Nobel Prize in 1974. Initially, it was believed that the lysosomes are nothing but a recycle bins or the garbage disposal units of the cell. But later on, with the advent of various kinds of research methodologies, various kinds of technological tools, now we know that lysosomes are very important cellular organelles, and they have a role in apoptosis, cell death, necrosis, and even in a cancer. So the lysosomes are such an important cellular organelles that are present in the cell. Now we will move to the next topic. When we talk about the size and shape of the lysosomes, we see that there is a no such a special kind of a shape that is present in a lysosomes, but the size of the lysosomes is very small, right? That is zero point 0.2 micrometer to the zero point 0.5 micrometer. Now, when we talk about the occurrence of the lysosomes, we have seen that the lysosomes are present in each and every eukaryotic cell. The number of these lysosomes varies according to the function of that cell. For example, you will find that in a plant cell, very few lysosomes are observed. Even in the fungal cells, you will find that very small amount of lysosomes are present. But when we talk about the immune cells or the phagocytic cells, for example, the neutrophils or the white blood cells, they have a numerous amount of lysosomes. So that depends upon the function of that particular cell. Now we will talk about the structure of lysosomes. These lysosomes are single membrane bound sac-like structures, and this single membrane encloses different types of proteins and different types of digestive enzymes. If we talk about the proteins, total proteins that are present in the lysosomes, there are 435 total proteins that are present in the lysosomes, out of which 100 proteins are present only in a lysosomal membrane. So you can imagine that what kind of composition that this particular sac-like structure has. Then what is the name of these enzymes? The name of these enzymes is acid hydrolases. Now, from the name, you can imagine that hydrolases means the enzyme which are bringing about the hydrolysis of a macromolecule. So, these and acid means that these particular enzymes, they will active in a acidic environment. So that's why they are called as a acid hydrolases. Now, how many enzymes are present? So there are different classes of enzymes that are present in the lysosomes. There are about 50 different enzymes that has been found in lysosomes, and these 50 enzymes, maybe they are nucleases, then lipases, then proteases. So all these kind of enzymes, they are present in the lysosomes. Now, why acid environment? See, we know that for a particular kind of a enzyme to be active, a particular kind of a environment is necessary. and that's why this compartment is there for a the name of the compartment is a lysosome. And you will find that in a lysosome, in that compartment, the H positive ion concentration is highest as compared to the cytosol. And this acid environment, uh, this acid environment favors the particular kind of a conformation of these acid hydrolytic enzymes or acid hydrolases, and this conformation is active, and that's why this acid environment is necessary. Then what will happen if a leakage is there in a lysosome? As I have told you that these acid hydrolases are present or they are active only in an acidic environment. That means their active site is active only in such a kind of an environment. But when they leak out of the lysosomes, they will come out of the lysosomes. They will come in a cytosol. So in a cytosol, we know that the pH of the cytosol is alkaline near about 7.2 or something. So that means here, in an alkaline environment, their conformation will change. So, acid hydrolysis will lose their active site, and because of that, they will not be able to act upon or digest the different macromolecules that are present in the cytosol. So, that's why even if there is a leakage, there will be no harm in a cytosol. But why the name suicide bags is given to the lysosomes? The suicide bags name is given to the lysosome, that is because when a large scale damage happens to the cell, or sometimes the lysosomes leakage simultaneously occurs, and because of that large amount of H positive ions they are set free out of the lysosomes and they comes in a cytosol, and they changes the pH of the cytosol. So because of that, when the pH is lower down, that means if pH becomes acidic, these acid hydrolysis they become active and they start to digest the whatever the cytosolic proteins that are present in the or cytosolic we can say the chemicals, whatever the molecules, macromolecules that are present in the cytosol that are digested by the these particular lysosomal enzymes, and that's why they are called as the suicide bags. Now, how this acidic environment is maintained inside the lysosome. So, you will see that there are two types of, there are two requirements actually. One is a proton pump. This proton pump is nothing but a V-type ATPs that are present in the lysosomal membrane, and ATP is required for this purpose. You can see here in the diagram, you will find that this membrane protein, this is nothing but a proton pump, and it utilizes the ATP and moves this H positive ions against their concentration gradient. So because of that, the cytosol is having 7.2 pH, but in lysosome or this in this compartment, you will get a pH of 5. So this is the interior of lysosomes, and because of that, the acid hydrolysis become active inside the lysosome. Now, lysosome, what happens when lysosomal enzymes they act upon the macromolecules? As I have told you that the large molecules that may be proteins, that may be nucleotides, that may be lipids or polysaccharides, all these are digested by the lysosomal enzymes, and they convert these macromolecules into their simplest forms. Proteins are converted into amino acids, the nucleotides are polynucleotides are converted into mononucleotides, the lipids are converted into fatty acids, and polysaccharides are converted into glucose molecules. That means their respective monomers are formed, and when these monomers are formed, these monomers are then transported into the cytosol. That may be this transportation, that may be because through the diffusion or with the help of certain kinds of transporters that are present in the lysosomal membrane. So this is how that the monomers which are there that are again utilized by the cell. So this is how the lysosomal digestion takes place. Now we will discuss special lysosomal membrane structural features. The first is more carbohydrates on a protein. Now we know that the lysosomal lumen is having acid hydrolases, and these acid hydrolases may act upon the proteins that are present on the membrane. Now, to save the proteins of the membrane, these proteins are more glycosylated. Now, the whatever the glucose molecules that are attached to these particular kind of proteins, and because of that, they form a kind of a layer of this glucose molecules, and or we can say the carbohydrate molecules, and that forms a glycocalyx. Now, such a kind of um, protective layer that is formed, and because of that, the lysosomal enzymes that cannot act upon the membrane proteins of the lysosomal membrane. So that is the most important feature of the membrane. Another feature of the membrane is a unique enzyme structure. As we know that all these hydrolytic enzymes, they are dangerous to each other. They are present in the lumen, and they may act upon each other, and by acting upon each other, they may cleave the enzymes. But the most vulnerable bonds of these enzymes on which the other proteins or other enzymes can act, such a vulnerable bonds are very well protected by their unique 3D structure. And because of that, the active site of another lysosomal enzyme cannot access this vulnerable bond that is present on the other enzyme, and because of that, because of this kind of a 3D structure, the enzymes cannot act upon each other. Then third most important structural feature is more lipids. Now we know that the what is the function of lysosomes? Lysosomes, the function is to fuse with the other vesicles. Now for that purpose, if it requires a fusion, or the more amount of lipid molecules is necessary for the fusion, and because of that, the membranes of the lysosome, they have more amount of lipids, and because of that, it is very easy for lysosomes to fuse with the other membranes. So these are the special lysosomal membrane structural features that we observe in the lysosomes. Now when we talk about types of lysosomes, see, there are three types of lysosomes. The first one is a primary lysosome. The lysosomes are formed by budding of a trans Golgi. Now, see here, this is a nucleus, this is a RER, that means rough endoplasmic reticulum, and this portion of the Golgi complex is called as a cis Golgi, and this portion of the Golgi complex is called as a trans Golgi. Now, from this trans Golgi, the vesicles bursts out, and these vesicles that contains a particular kind of enzymes, or a special kind of enzymes, these are called as acid hydrolases, and these acid hydrolase containing vesicle, that is called as a primary lysosome. Now, this primary lysosome does not have acidic environment. Now that's why we call it as a primary lysosome. So whatever the acid hydrolases that are present in these primary lysosomes, they are inactive. So this is called as a primary lysosome. Then another is a secondary lysosome. So when these lysosomes, these primary lysosomes, they fuses with the food vacuole, as they fuses with the fluid vacuole, they start to accumulate H positive ions, and because of that, or they convert the internal lumen of this secondary lysosome into acidic environment, and because of that, now the whatever the acid hydrolases, they become active, and they start digesting the food material that is present in that vacuole. So this is known as a secondary lysosome. Now, after, you can see here, after digestion, the third type of lysosome that emerges, and that is known as a residual body. And this residual body contains the undigested food material. This undigested food material is expelled out of the body by exocytosis. So this is a third type of a lysosome, which is called as a residual body. So this is how there are the three types of lysosomes that we see in a cell. Now we will see functions of lysosomes. The first function of lysosomes is the intracellular digestion of food. As I have told you that the food particles, when they come in the vicinity of amoeba-like organisms or unicellular organisms, these food particle that is engulfed by these organism with the help of a pseudopodia, and it forms a food vacuole, and this food vacuole fuses with the lysosomal vesicle, and the whatever the food material that is present in that food vacuole, that is digested by the lysosomes. Now, the different types of lysosomal enzymes, they act upon this food vacuole and convert that macromolecules into monomers, and these monomers are absorbed by the cytosol. So that the monomers that are formed, they are transferred to the cytosol with the help of a transport protein molecule, or they simply diffuse out of the lysosomes, and whatever the residual material, undigested material, that is thrown out of the cell. So this type of we can say the food particle digestion, that is called as a phagocytosis or a intracellular digestion, because food is taken inside the cell and then it is digested. So that's why it is called as a intracellular digestion. And these lysosomal vesicles, they help in digestion of such kind of food vacuole. So this kind of digestion is seen in very primitive organisms, unicellular organisms, and you can say the choanocytes. You must have heard about this. The choanocytes that are present in the poriferans. So that is the first function of lysosome, that is intracellular digestion of food. Now the second function of um, lysosomes is extracellular digestion. As we know that the sperm acrosome contains hydrolytic enzymes. These are nothing but the lysosomal enzymes, and these lysosomal enzymes, they are helpful in sperm to reach up to the ovum. So the ovum is surrounded by a cumulus mass, and that is made up of a kind of a polysaccharide. So the enzyme present in the lysosomes, that is called as a hyaluronidase, that enzyme helps in digestion of this cumulus mass, and it creates a kind of a tunnel through which the sperm reaches up to the ovum and fuses with the ovum. So for a fertilization, the lysosomes, they assist in uh, the fertilization of a sperm with the ovum. So that is extracellular digestion. The third function of lysosomes is phagocytosis. In Greek, phagein means to eat and kaitos means a vessel. So this is a kind of a eating vessel. When a microbe, this may be a bacteria or a virus, when approaches to the WBCs, or we can say macrophages or neutrophils, these WBCs or neutrophils, they are antigen presenting cells. So they have extra kind of a function here. Apart from the phagocytosis, they also show antigen presentation. So you will find that the microbes that are present in the vicinity, they are engulfed by these cells, and the vesicle that is formed fuses with the lysosome, and the lysosomal enzymes act upon this, and it digests all the material that is present in the microbe, or we can say they first kill that microbe by digesting the plasma membrane, and then they eat it out. So whatever the monomers that are formed due to the digestion, that are absorbed simply by diffusion, or with the help of certain kinds of transport protein that are present on the lysosomal membrane. These transport proteins, they help in taking up that monomers from the lysosomal human to the cytosol. Apart from this, the parts of this microbe that are also displayed on the MSC molecules by these cells, so that can help in immune um, stimulation. So such a kind of phagocytosis that is seen in white blood cells. So this is about the phagocytosis. See, now we will discuss about the fourth function of lysosome, that is autophagy. Auto means self, phagy means to eat. Now, when cell starts to eat its own organelle, then it is the phenomenon is called as a autophagy. Now, how it eats self organelles, that is with the help of lysosomes. Lysosomes fuses with the worn-out cells. Now, why lysosome fuses with the worn-out cells? We know that there are different types of cellular organelles, and they have a certain period of lifespan. Now, for example, we know that the RBCs have 120 days. Even in case of mitochondria, the liver mitochondria, they have a lifespan of 10 days. So, such worn out cells, they no longer useful for the cell. So, such a worn out cellular organelles, these cellular organelles, they are diffuses with the lysosomes, and they are digested, and the whatever the material, or we can say the monomers, after digestion they are formed. These monomers are then absorbed in a cytosol. So this condition is found during the starvation of a cell. So for a period of a time, certain period of a time, these lysosomes fuses with the cellular component, and they devour that or eat that cellular organelles. So this is known as a autophagy. Now, you know that the in some cases also, if the cell is not functioning well, in that case also, or a malfunctioning cell also shows such kind of a autophagy. So that is the reason. Now you can see here how this happens. Here, this is a, we have seen this diagram before also. This is a RER, this is Golgi and trans Golgi. It produces a lysosome. Now, this is the worn out mitochondria. This worn out mitochondria, around this worn out mitochondria, a kind of a double walled cellular membrane is created. Now, how this cellular double walled membrane is created, that is not known. But this particular structure is called as a phagophore. Now, this phagophore, when this phagophore forms this, it has a special structure. It is enclosed in a double-walled membrane. So, this one is outer membrane, and this one is a inner membrane. Now, this lysosome fuses with the phagophore, and it the fusion is with the outer membrane of the phagophore. So the, you can see here the green colored membrane is also being digested here. So that means this green color means inner membrane. So along with the worn out cellular organelle and this inner membrane, this total cellular organelle is digested, and monomers that diffuse out of the this autophagolysosome or autophagosome, and these materials, or there may be certain types of monomers that are transported to the cytosol. So this is how that whatever the mm, uh, monomers that are present in this worn-out mitochondria that are again recycled in the cell. So this is known as a autophagy. Now, the fifth function of lysosomes is the lysosomal pathway of apoptosis. And we know that the lysosomes have also seen taking part in selective degeneration of organs or cells during the metamorphosis of insects and amphibians. So this pathway is called as a lysosomal pathway of apoptosis, and this occurs due to certain kinds of release of certain kinds of proteins from the lysosomal membrane. That is because of the extra, or we can say increased lysosomal membrane permeability, that is called as a LMP. And this triggers the release of certain kinds of protein. Here that protein is called as a cathepsin proteases, and these cathepsin proteases, they are responsible for triggering the uh, apoptosis and cell death. So this is known as the lysosomal pathway of apoptosis. Now the sixth function of lysosomes is its role in a rare disease that is known as a lysosomal storage disease. Here we know that lysosomes are having acid hydrolysis or the enzymes, and these enzymes are made up of a protein. Now these proteins are synthesized by the instruction of a genes. Now if there is a mutation in a genes, what will happen? The there may be the absence of that particular digestive enzyme, or there may be even if that enzyme is synthesized, that may be inactive. So because of that, the instead of digesting this particular macromolecule or a respective macromolecule, the lysosome will start to store that undigested macromolecule, and this is known as a lysosomal storage disease or LSD. Now, more than 50 such type of diseases have been identified in human beings. One of them is the Tay-Sachs disease, or there are a number of diseases. Pompei's disease, that is also a kind of a lysosomal storage disease. In case of a Tay-Sachs disease, uh, the undigested macromolecule is a lipid. So the lipid digesting enzymes are not present in the lysosomal repertoire, and you will find that these lipids will start to accumulate in the brain cells, and that will manifest into a particular kind of a disease. So that is called as a Tay-Sachs disease, where the cells of the uh, brain, they malfunction. So this is the role of LSDs, or this is the role of lysosomes in LSDs. Now there are certain functions of lysosomes are also there. That is, lysosomes also take part in iron homeostasis. They are also helpful in activation of inflammasomes. So these are the functions of lysosomes. So if you like my video, please subscribe to my YouTube channel, BAMU'S Biogenius. Thank you very much for patient hearing.